Thank you all for being here today. I want to thank the TAGC selection committee for allowing me to share this with you. I'm going to be talking about how we used a zebrafish model to functionally validate an albinism allele that we discovered in a Caribbean Native American population. So I want to start by acknowledging all of my lab members, all of our collaborators here, and then obviously our funding sources, but I particularly want to acknowledge the Kalinago community in the island of Dominica for their help with this project. Without them, this would not have been possible. Here you can see Dr. Ang here and our collaborator John Hawley pictured with the Kalinago Council. That's the governing body of the Kalinago territory. So to go into some of the motivation behind our work and how we discover this polymorphism, our lab is interested in the primary genetic basis of East Asian and Native American skin hypopigmentation. So the alleles responsible for European hypopigmentation have been discovered. Those are SLC 24A5A111T and SLC 45A2L374F. Those are largely responsible for the hypopigmentation seen in European populations. But the corresponding allele or alleles in Native American and East Asian populations have not yet been discovered. And the reason we're interested in finding these alleles is because there's quite a difference in melanoma susceptibility between populations. So European populations are actually around 20 times more likely to develop sun-induced melanoma compared to both African and East Asian populations. And so despite East Asian and European populations having similarly light skin pigmentation, there's quite a difference in melanoma susceptibility. And so we think by finding the primary alleles responsible for this hypopigmentation, we can, that'll be a great first step towards finding the genetic underpinnings of this difference in susceptibility. So a great way to look into this question is to use an admixed population. Since we're interested in East Asian skin hypopigmentation compared to the ancestral African phenotype, we used the Kalinago population, who has primarily Native American and African genetic ancestry. They also have a wide variety of skin pigmentation phenotypes, which we measure by melanin index. And that's actually a quantitative measurement of skin pigmentation using skin reflectance readings in the underarm. And we can then correlate that with ancestry and also compare to certain candidate polymorphisms that we have. And here you can see the amount of Native American ancestry in this population is negatively correlated with the melanin index in these individuals. So while we were there collecting samples, we found that there were 3 individuals with albinism in this population. You can see one of these individuals here. He's pictured with his 2 parents and 2 siblings that you can see do not have the albinism phenotype. The 3 individuals with albinism had light hair, light eyes, light skin, and nystagmus. And so we initially genotyped these 3 individuals and compared that to 28 previously reported variants that have been found in Native American and African populations. They had none of the previously reported variants. And since we were doing analysis comparing melanin index and genotype, we knew that we'd have to control for whatever variant was responsible for albinism in this population. So to figure out what variant was responsible, we did whole exome sequencing on one of the albino individuals and one of their parents, who is an obligate carrier. And we found variants that were homozygous derived in the albino individual and heterozygous in the parent. 2 of these variants are both of them were found in the oculocutaneous albinism 2 gene. They're both missense variants. The first is a SNP, R305W, and the second is a 4-nucleotide inversion, NW273KV. And upon genotyping around 450 individuals, we found that only the 3 individuals with albinism were homozygous derived for the NW273KV genotype. So as I mentioned, the NW273KV is a 4-nucleotide inversion. It's located within an African haplotype in these individuals. And here's another melanin index chart. The top is what we call triple ancestral. So they're homozygous ancestral for those 2 European alleles that I mentioned previously, and then also for the NW273KV polymorphism. And you can see they have a lower melanin index range of values here, and here are the 3 individuals with albinism that had the lowest M-index values in the entire population. So what does OCA2 do normally? Its primary role is to encode the P protein, which has been shown to be an integral membrane protein, or integral protein within the chloride transporter channel within the melanosome membrane. Its primary function is to reduce the conductivity of the membrane, which inhibits the activity of VATPase, which lowers the acidity of the melanosomes and ultimately leads to melanin production. And so since our polymorphism is changing an NW to a KV, the N in the NW has been shown to be an N-glycosylation site, which could affect protein folding and stability. This change, removing that site is likely the causative mechanism for how this polymorphism affects melanin production. So we think we have the variant, but we want to functionally validate that. And so we worked with in vivo biosystems to generate a CRISPR-Cas9 knock-in construct. We did the same amino acid change, NW to KV. We injected the embryos at the single cell stage. And here you can see the adult F0 mosaic crispant with the patches of albinism here. We then crossed these F0 crispant fish, which produced, here you can see a 2-day-old albino F1 fish, so a complete loss of pigment. And then here's one of these fish also as an adult. So this is an initial qualitative confirmation. We see a lack of the pigment development. But to be quantitatively certain, we did allele-specific PCR. And here you can see we see the expected bands for the amplicons associated with ASPCR, and we don't see any for the negative control wild-type fish. We also saw some expected variability in the F1 phenotype. So as I showed before, there's the albinism phenotype. We also have some of the F1 fish that appear almost wild-type in appearance, which we refer to as dark. But then we also see an intermediate phenotype, somewhat in between these two, where you can see the melanophores are slightly less pigmented. They're a bit more transparent. But you can also notice that the intermediate fish have the dark eye, similar to the dark or wild-type fish, while the albino fish lose that pigment in their eye. This difference in the dark and intermediate fish persists into adulthood. You can again see a qualitative difference between the adult dark and intermediate fish here. And if you compare the regions of pigment development, you can see the melanophores are reduced in size and number. And then if you compare that to the adult albino fish, you can see there's a total lack of pigment development in those areas. So I don't think I have to emphasize the importance of using zebrafish to model human diseases at a meeting like this, but I want to show the importance that this demonstrates for using zebrafish to model skin pigment diseases in particular. So as I mentioned before, we have the 3 individuals with albinism in the Kalinago community, also all had nystagmus. So that's something that we're interested in looking into further in these fish as an additional phenotype. And a great way that we can do that, another common technique that we use in our lab is micro-CT. So here you can see on the top left is a micro-CT Z-stack of a 5-day-old fish stained with PTA. And the region I've highlighted here is the optic chiasm. And so given that these individuals in the Kalinago population have nystagmus, we're interested in seeing if there's any phenotypes associated with the eye, the retinal development, the optic nerve, and seeing if there's any phenotype associated in the zebrafish as well. The image on the bottom here is actually just a separate line that we have in our lab. That's a transgenic line that expresses GFP in its retinal ganglion cells. So that's just for reference here of where the optic chiasm is and what I'm indicating in this box up here. And then we can also look into other phenotypes associated with not just this allele, but with other alleles. So if you remember, there were the 3 individuals with albinism were the only ones who were homozygous derived for the NW273KV genotype, but there were 5 individuals who were homozygous derived for the R305W genotype. So 2 individuals that did not have albinism were also homozygous derived for this allele. So we're interested in there potentially being a separate phenotype associated with this polymorphism. So a CRISPR knock-in is something that we're planning to do with this as well to see if there's a distinct phenotype. So in summary, we discovered this polymorphism, OCA2 NW273KV, in a Caribbean Native American population. We knocked this into a zebrafish model. We qualitatively confirmed with the albinism phenotype, and then quantitatively confirmed with ASPCR. Collectively, this functionally validated this polymorphism as responsible for albinism in this population. And then future work could yield insights into additional phenotypes associated with this polymorphism or phenotypes associated with additional polymorphisms. And with that, I'll take any questions.
Very nice, thank you. Were only the melanocytes affected, or did you also see an effect, for example, in the iridophores?
So we haven't looked into that directly, but we do have stains developed for certain pigment cells with micro-CT, so that's something that we could also do to see if there's a change in the other pigment cells as well.
Hi, great. Is the color variation heritable, or is it remain variable across generations? If you breed two light ones together, are all their kids light, or is it variable again?
So we haven't gotten that far yet. These were just taken a couple weeks ago, so we're planning to do that soon. Yep.
All right. If there are no other questions, please join me in thanking Thaddeus. And also, how about a big round of applause for all of our speakers for a fantastic session. Thank you very much.